Is everything fine, Mr. Frost?
Everything fine.
Okay?
You can start right away.
Okay, then thank you very much for your kind introduction. I welcome you all to our smart webinar series today. The first webinar has the title BioRam, Raman Trapping Microscopy, Next Generation of Single Cell Analysis and Cell Sorting. Okay, now I have to find out how I can change my foils. Okay, it's a little bit tricky. I do my very best. I'm sorry. Okay, first, today's schedule. First, I want to provide you a short overview about the current cell analysis methods and their drawbacks. Next, I want to provide you an introduction into how Raman spectroscopy basically works with a short excursus to laser trapping. And of course, I want to tell you or give you some of our applications, what we already performed so far with our Raman spectroscope. And last but not least, of course, there will be a short summary. I think you all know that there are a lot of different biomarkers for cell analysis out there. However, most of these biomarkers depend on the usage of antibodies, which are often no, with or other labeling techniques. And in case of antibodies, the first problem is that there are often no good antibodies for the marker of interest. In addition, cell staining is often associated with cost and time-intensive preparations, and a further disadvantage of these preparation procedures is that the antibodies or staining agents can be very stressful for living cells. So cells might change their characteristics, then cells may differentiate, and I think as you can see on these representative pictures here, the cells are not really happy if you overload them with antibodies and stainings. Last but not least, of course, labeled cells are so far not allowed for therapy of patients. So all in all, there are many different limitations, and our question was, of course, is there a possibility to overcome these limitations with another technology? And with this question in mind, we from Cell2 left the fields of biology towards other academic fields like chemistry and physics. And there we found that it is really possible to analyze different materials like metals, alloys, powders with nothing else but laser light. And so we thought about, hey, maybe we can use this technology also for our biological questions. And the technique I'm talking about is the so-called Raman spectroscopy. It is based on the Raman effect, which was found in 1928 by Mr. Raman. And although this sounds like, oh my God, it's maths and physics, I can assure you that this method is not that difficult. What basically happens is that you put your cell under a microscope and then you turn the laser light on. We use a laser of 785 nanometers, as this is a wavelength which shows to not harm the cells in any way. And by traveling through the cells, the light photons interact with the cell molecules. And the majority of the photons pass the cells without getting disturbed. However, a small part of the laser photons interact with the atomic bindings in the cell molecules in a way that they transfer some of their own energy to these bondings and thereby lose a part of their own energy. And with this, they got a redshift in their wavelength. This is the so-called Stokes scattering. And these Raman photons, we detect then with a specific spectrograph. As each molecule leads to a different change in wavelength, in the end you end up with a Raman profile like this. And just that you don't be confused, in case of Raman analysis, the wavelengths are not called wavelengths, but it's called wave numbers, which is just the reciprocal of wavelength. And here are examples for two molecules, in this case the amino acids phenylalanine and cysteine. So you see each and every molecule has its own specific Raman spectrum. And you can imagine all the different molecules within the laser focus, the analyzed part of the cell, contribute to the Raman spectrum, and altogether this forms a fingerprint which is, or a Raman spectrum which is as specific as a fingerprint for cell analysis, and with this you can discriminate cells or detect cells. So far about the theory. Now let's have a look on how Raman spectroscopy and cell analysis can be combined. And for this, we established our so-called BioRam device. It is basically an inverted microscope with a Raman unit. And it's a benchtop device which is really built up so that biologists and physicians can easily use this specific technology. It has an inverted microscope, as I said, so that you can analyze your samples without any contaminations. And in addition, you have different sample holders for multi-well plates, for slides, or also for small dishes, so that you can use your consumables you always use. The only thing you have to take care of is that the consumable bottom needs to be of glass, not of plastic, because plastic itself has the software. The system allows to analyze your cells in real time and navigate through your samples. You can put little pins in the part of the cells you want to analyze, and as you have subcellular resolution, so basically our focus is around 2 micrometers as a sphere. You can, for example, discriminate between cytoplasm and nuclei even. And in the end, what you get, these are the raw Raman profiles, as you got them from the software. However, in order to really analyze these data, you have to pre-process the spectra. You have to, for example, remove the background and do also some peak corrections. However, all these mathematical algorithms you need for pre-processing the spectra are already implemented in the software. And after doing this, you get Raman profiles like this. What you see here are Raman profiles of erythrocytes and thrombocytes. And in both cases, there is an overlay of around 30 Raman profiles. So each of these small lines is one Raman spectrum. And you see here the overlay in this plot. And I think you can already see per eye that there are really differences between the erythrocytes and the thrombocytes, for example, here at 1,200 wave numbers. However, it's not always that easy to see the differences between cells or cell states. And so we also use another mathematical methodology to analyze our data. This is the so-called principal component analysis. And the principal component analysis, or short also PCA, is simply spoken an algorithm which is looking for the largest differences between data points. And in this plot you see the spectra of erythrocytes and thrombocytes again. Here is an overlay of the mean spectra with standard deviations. And using the PCA, you get a dot plot where the spectral data are transferred in these, where's my mouse, in these plots. So each single spot is one Raman spectrum. And the scores plot shows you how a single spectra is different or similar to all other spectra. Beside the scores plot, you get the so-called loading plots, in which you see which part of the Raman spectra are most important for the distribution of the data within the scores plot. The larger the peaks in the loadings plot, thereby doesn't matter if positive or negative, the more important this part of the spectra is for the distribution of the scores here. Another thing is that specific parts of the Raman spectra are associated with different biomolecule groups like DNA, RNA, proteins, and so on. And so you cannot only learn if the cells are different or not, but you can also see from this analysis which biomolecules contribute to the difference of different cells. So much about the theory of Raman analysis. Next I want to tell you about another feature of the BioRam device. When you want to analyze single cells with Raman analysis, it is really important that the cells stay in the position during the analysis. And this is especially important if you have small cells like bacteria or small particles which float around very fast. And the sophisticated setup of the BioRam device ensures that also such small particles can be analyzed without any problems. As the so-called optical trap builds on a force field around the cell, which is analyzed, thereby holding it in place during analysis. And I hope this video works, because here you can see how the Raman or the laser trapping works. We turn on the laser, and now we focus up in Z direction, and you see all the other cells which are not analyzed blend into unsharpness, whereas the one cell is still sharp and can now be moved from one well to the other. And as soon as we turn off the laser, the cell sinks down again to the bottom. Now, I told you a lot about the analysis and the device in general, but in the end, of course, the question is, what can you really do with this method? And speaking about applications, I can assure you that they are really versatile, including, for example, tumor cell analysis, drug screening, regenerative medicine, differentiation of stem cells, and of course also quality control. And now I want to show you 4 of our applications we performed from our experiments we performed so far to give you a short insight into this versatility. In the first example, I want to show you the analysis of bacteria. Together with one of our cooperation partners in Würzburg, we had a look on Staphylococcus aureus and Pseudomonas aeruginosa. We got the samples from different patients infected either with Pseudomonas or Staphylococcus, and we cultured and diluted these samples several times to really get pure strains for Raman analysis. As you can see here very well, in the scores plot, we could really well discriminate the Pseudomonas from the Staphylococcus aureus bacteria. Furthermore, if we had a closer look, we found also that we can not only discriminate the Staphylococcus from the Pseudomonas, but also different substrains within the bacteria. So from different patients, we could discriminate different substrains. And of course, we also had a look on the loading plots to find out which part of the which biomolecules are important for discrimination. And in case of Pseudomonas, it was mainly the collagen and lipid content, whereas in case of Staphylococcus aureus, it was mainly RNA and proline content. And thinking about the fields of application, what you can do with this, of course, you could, for example, use Raman analysis for epidemiologic studies to analyze, for example, infection relapse and also test for treatment resistance, and of course for the identification of microorganisms. In a second experiment, we wanted to answer the question if it's possible to detect nanoparticles within the cells. And to do so, together with a cooperation partner from London, we used human neural stem cell line, CTX-OE03, and we incubated them with iron nanoparticles. And after several hours of incubation, we fixed these cells in PFA and compared them with other CTX cells which were not incubated in these nanoparticles. And you can see here representative pictures. So the cells look more or less the same. However, you can see here these dark spots. These are the iron nanoparticles. And if you have a look on the Raman spectra, you see here again mean Raman spectra with standard deviations, especially in this part here, the cells treated with nanoparticles show a really different spectrum, whereas these parts, these three peaks here, could also in literature be associated with magnetite and manganite, and which are both iron oxides. So what we see here are indeed the iron nanoparticles within the cells. And this could be also very well be detected in the scores plot. Here you see the background, the control cells, and a broad amount of cells which are treated with nanoparticles, whereas the more the cells go to the right, the more nanoparticles they included. And also in the PCA loadings, it can be clearly seen that these three peaks, which are also visible in the Raman spectra themselves, are most important for sample discrimination. So with this we could show that we can detect really nanoparticles within the cells, and we are very optimistic that we not only can detect iron nanoparticles but also other nanoparticles, as well as, for example, microvesicles and also bacteria and viruses. In the next experiment, I want to show you some of our first data of our current project in which we want to improve the quality control of blood products by providing a faster, easier, and really highly reliable test for quality control of each and every blood product. And what you see here are thrombocyte samples. We received them in such bags, and we analyzed them every day for a time course of 14 days. And you can see here representative pictures of different days, and you can see here very well that on day 2 the cells look like, let's say, normal thrombocytes. There are single cells, and the older they get, sorry, the more they agglomerate and lose their function. Having a look now on the Raman spectra over time, we found that they are more or less similar, which is not that unexpected as it's the same cell line or the same cell type. But there was one part in the spectrum which is the peak of, or at 1050 wave numbers, which decreased over time. So as the cells got old, this peak decreases. And as I said, we analyzed not only 4, we analyzed 8 donors, and in each of this donor, this change in Raman spectra could be detected. This is shown here. Here you see the PC2 scores over time. The PC2 scores, it's that because of the peak at 1050 wave numbers was then found later in the PCA on PC2. And you see that the values change over time. And not to be confused, they got higher because the peak was negative correlated with PC2. That is why the scores got higher, although the peak is getting lower. And we're really happy with these first data, and of course we will do a lot of more work to really show that Raman analysis can be used for quality control of blood products. And of course you cannot use Raman spectroscopy only for blood products, but of course also for quality control of other cell-based products. Last but not least, I want to show you another experimental setup, which is very neat. What we did here, we used more than 13 cells and treated them with doxorubicin, which is a known cell-killing agent. This alone is not really special. the most special thing is that we used in this experiment the so-called microwells. And I think you can see here very well. This is the slide, and on this slide are these small microwells. And we seeded the cells in a way that more or less each microwell contained one single cell. And as the BioRam technology or the BioRam device remembers each and every spot where it took a measurement, you can go back to the spot and analyze the one single cell again and again. And this you can see here. We analyze the cells at 0 minutes, then after 10 minutes of doxorubicin application, and after 1.5 hours. And you can already see that the cells start to disintegrate, they start to die over time. And this we could also detect it by using Raman spectra alone. Here again you see the scores plot, and you see the, there's my mouse, you see here the living cells, and then after early apoptotic and late apoptotic cells. And what you see here in this experiment also very well is that it really makes sense to look on single cell basis, because not all the cells behave the same way. So there are some of the cells which already were in a late state of apoptotic already after 10 minutes. And using especially these microwells, it is really a good possibility to monitor living cells over time, also cells which are in liquid and not adhesive on a plate. With this, I'm already at the end of my talk. As I said, I only provided you with a few examples of all the things we already analyzed. And as first take-home message, I want you to please keep in mind that Raman analysis is a fast and easy to use, label-free and non-invasive method, which allows you a totally new view on your cells. And if you are interested in analyzing, for example, your own cells, you can either send us cells to our service lab, or you can come together with yourselves and analyze some samples for yourself. In both cases, I would be very happy if you just contact me, write a mail, or just call me. And now I told you a lot about what you can do with Raman analysis in case of single cell analysis. Now I will give over to my colleague, Mrs. Karin Schütze, who will talk about what can you do with Raman spectroscopy in case of 3D tissues and also theories. So stay tuned and have fun with the second seminar.
Heidi, thanks a million for giving us this wonderful introduction. I think a few questions came up, and I think there might be 1 or 2 minutes time to answer perhaps a few of them. One question was I think came in twice. What is the power of the laser you are using in your system?
The power of the laser that we use is a maximum of 80 milliwatts, but it's now focused through the laser and system. Only a few amounts of this arrives at the laser beam. Sorry, this is Karin. I took over already for Heidi.
Okay. Sorry, sometimes we have some, I think, problems with the sound. I have no idea why. Another question came in. Will fixation with ethanol change the Raman spectrum?
We tested different kind of fixation methods, and we realized that the paraformaldehyde is the most preserving for the metabolome. So I recommend to use this, and ethanol would really destroy some parts of the information.
Okay, and then one general question was, can you briefly mention the limitations of your technology, if there are any?
The limitations of the technology would be about, in that case what I'm talking now about, that samples would be too thick, like millimeter or so. So usually we go up to 500 micrometers. The limitations regarding the cells is really difficult to say. We have tested at least 100 different kind of cell cultures and so on, and in most of the cases we get some information. We have to take care, however, that the substrate is possible to work biocompatible, or let's say Raman compatible, because some of the scaffolds that usually are used may hinder this. So this would be one of the difficulties or challenges that we need to face.
Okay, perfect. All the other questions which came in and will come in, we will answer at the end of the webinar, also by email, so don't worry. I think all the answers to all your questions will be in your inbox, I think, this afternoon. Okay, then we can continue.
Okay, now welcome to the second part. My name is Karin Schütze, and I would like to introduce to you shortly, for those who have not been there for the first time, why do we use the Raman analysis? It is because we get the information about the entire cells and not only about the surfaces. And Raman is well known in any,
Can you
still hear me?
Yes, now it's okay. Now we have full size of the presentation. Everything fine.
Okay. Yeah, the question was, why can we still look into our cells when we have such a high amount of molecules that are undergoing a permanent changeover? And this was what everybody was really wondering, that it works, that we can use the Raman, which is focused to about a spot size of a micron towards the cells, and measure the information that we get from the interior of the cells, not only, as usual, from the surface. And that's what makes it very interesting, because molecules within the cells always change as soon as anything changes in the body, like if cells change towards development, if they got diseased, or if there are any drugs or toxins. And this makes the system or the application of Raman's microscope so special. And the good thing is that we only need a small sample amounts and not dependent on any kind of antibody, which also sometimes improves the application. This again is the system, and as you already heard, we can focus to small spot size, so we can work subcellular even, we can compare a nuclei with the surrounding cytoplasm. We see difference in the spectra, and we learn what makes the difference between the two kinds of spectra, and we use statistical analysis, very normal ones like the principal component analysis, very well known in biomedical applications. And we even learn what are the molecules that are important for the differences. Not in all cases, but in some cases we can find such information. CellTool has a couple years now working also on the biomedical field and learned a lot of applications, and also we got extensive expertise how to interpret the statistical data to a meaningful analysis. And as you know, we can also make customized analysis, and we also have a demo lab so that you can come and make your own samples. What is unique is that we also combine the Raman spectroscopy with optical trapping. That means simultaneously, as soon as we switch on the Raman laser, immediately the trapping starts, so that you can work even with cells in solution. They don't float away while you do measurements. And we have started in several different applications, like for example here in tissue sections, but also in cell cultures and drug screening within single cells, but also in 3D tissues, as I'm going now to talk a little more in detail. Looking into tumor spheroids is currently a very interesting method to study drugs, as this imitates much more likely what's happening in the body as compared to 2D cell systems. So what we could show that we can go into spheroids, look in the equator line or go into the Z direction and still get spectra from any places that we look in. And so we can either look up to 300 microns in this case. It depends a little on the kind of tissue, the opaqueness. So sometimes you can go even up to 1 millimeter into the tissue, for example, in brain tissue. And the idea is now, instead of what is currently done, take these spheroids, fix them, cut them into slices and do the routine tissue analysis. We just go into the living example, show how drugs, for example, go inside from the side, how they penetrate, to see other drugs already arriving in the center part, or are they still only at the outside, which gives a lot of help, especially in the drug developmental part of the pharmaceutical industry, to see how their cocktails react on the real or the more likely situation in the body. We also could show a combination of tumor spheroids that have been in co-culture with fibroblasts. In this case, this all is in a hydrogel. So the hydrogel is perfectly suitable for Raman. And what you can see is if we look into the fibroblasts without co-culture, we see a certain spectra. And when we look into them in co-culture with this tumor spheroid, something happens. And we can clearly see this in the Raman spectra. Here is displayed at the score plot. And if we look into the main peaks, which makes the differences between the fibroblasts in co-culture as compared to non-co-culture, we see the color seems to change. And if you have a careful look into the microscope, we can also get the idea that the co-cultured fibroblasts seem to be much more denser, meaning they have obviously built up a higher collagen network. So this was very nicely to see, and especially here we can use the hydrogel and also penetrate hydrogel with drugs to follow how those cells react, because this is a very flexible gel, and this is very good to be used in combination with Raman spectroscopy. Another structure that we have worked on is the so-called mucoderm, which is a pretty tight structure. We tried to see how deep can we go in here. So as you can see here in the beginning, we still see some microscopic resolution, but then we don't see anything anymore in details, but we get still very nice spectra that almost did not change even from the height as compared to the beginning in the 0 or 20 micrometer distance from the bottom. And the good thing is that if you have now any kind of fibroblasts or cells within the so-called mucoderm, we can on the one hand differentiate between mucoderm and cells, and we can even differentiate cells with Raman spectroscopy that have been treated or untreated, which makes it a very helpful tool to look into 3D and to still differentiate, in this case, activated fibroblasts compared to the non-activated one. And as you see, we only take a few cells. For statistical reasons, we would recommend to take maybe 300, 400 cells. But this is enough to get information how cells react. This is nothing compared to the routine FACS analysis, where you usually need a lot of material before you can get started. And of course, in this case of a 3D structure, it would be difficult to get the antibodies into it. So this is something where we think Raman spectroscopy might be a big help for future analysis of 3D tissue or cell-based products. And talking about cell-based products, this is a project that we have a lot of experience now ongoing. This is a collaboration with the tissue biology unit in Zurich, and the challenge is to have burns or navels or scars. There people would like to help them, and what they do is they take a piece of healthy skin, about a 2 by 2 centimeter part. They take these and expand them in differentiation mediums so that only keratinocytes grow in one place, and in the other only fibroblasts grow. So they have two types of cells. Here it is important now to make quality control before these cells are applied to the final graft. How do they make the final graft? They take the fibroblasts and mix it with a gel, let it grow about 5 days in the gel, and then on top they put the keratinocytes on top, so you have a real skin graft. Also here now it is necessary to make sure that this product is safe before it is applied to the patient. So this 8 by 8 centimeter graft beautifully grows on top of a wound, and it also heals very, very successfully. Now, when we look about the current used methods, it is FACS analysis that they use to make sure there is no cross-contamination in those two cultures, and there's no melanocyte in there. But this takes time, and the antibodies for fibroblasts are not very good, and it would be also interesting to reduce costs and get the results much faster. So what we tried is in the first set, can we discriminate between keratinocytes and fibroblasts in culture, and in case there would be a melanocyte, would we be able to identify this with a Raman spectroscopy? And you can see these are the melanocytes. They are completely different as compared to the fibroblasts and keratinocytes. And when we look now into into the analysis, how can we make sure that we get only less than 5% cross-contamination? That is what Swissmedic asked for. We had to develop a different kind of analysis. In this case, we use a so-called cluster analysis. And what we do is we just take all the cells that we have analyzed, 400 cells, to measure how many different clusters do we get. And we got 3 different clusters. You see how many cells we have. These 155 and the 224 cells, and we looked into the spectra and we saw they are very similar. So it seems that we have a different cell stage that we measured here, but here we got some spectra that we could not align to. So this would be the contamination in the cells, and the same thing we did with the fibroblasts, and we also saw here similar spectra regarding their developmental states, but no other kind of cells. So when this is safe, then they start to make the product, and with the product we can now put the Raman system on top, which are the keratinocytes layer, and we measured automatically running through here a so-called scan. So every 10 microns we make a measurement, and automatically the numbers are taken. If we look into the graft, here we have fibers and we have fibroblasts. And then we can now measure within the depth of about 500 micrometers. And what we can see is that the spectra of the keratinocytes differ now much more in the 3D structure. That's due to the different kind of fibroblasts that usually also get elongated. And if you look now by accident in an old graft and a new graft, we could see the differences, which makes us very confident that we see DNA breaks when we have old grafts that have been 2 weeks, for example, in this case, on customs. So we can not only measure how many cells are there and measure whether they are still, if there's a cross-contamination, but we can also measure whether the cells are still intact and functioning. And also here we wanted to look whether there's a contamination with melanocytes, and we did not find any contamination of melanocytes. And you see the number of cells we've analyzed is very small, so we don't need to grow up a lot of cells. We just work directly with the system, and we don't, with the graft, and we don't need to destroy it. And as you can see here in the keratinocytes, which is on the top layer, we found these kind of spectra which are similar due to stage, but then we found a different kind of spectra. And when we compared this, we saw these correspond to fibroblasts. And then and that is the good thing about our system, we always make images or the pictures from each of the measured areas. And when we looked again, we saw that the fibroblasts, in this case, were really sneaking through the surface of keratinocytes. So we could identify the area where we had fibroblasts. But this, of course, is no contamination, and we could go further on, and they can use the system now further for the patient. And altogether, we realized that using Raman spectroscopy, there's a lot of savings. On the one hand, on the cells expansion time. Also, we could give much more information within 4 hours as compared to 48 hours as they used for the normal FACS analysis. And if you look into the graph, the engineered graph, you could see here they use in the moment DNA count, which gives them only the idea of how many cells are there. But with our system, we could differentiate between what kind of cells are there and is there any kind of contamination, and are they still functional? Which makes us very confident that we will get together with our collaborators the GMP validation for this kind of cell-based product. Another thing that we recently have done is to look into zebrafish embryos. The question was, is it possible to go even into entire embryos and measure within certain tissue areas, and can we see differences with them? And also here we evaluate different kind of fixation methods. What we could see is that different fixation methods really matter. It's also important that you wash it very carefully with PBS, for example, and that glutaraldehyde gives different signals as compared to paraformaldehyde fixed cells. And what we also did, we looked into the 24 hours and 72 hours embryos, which means these are 2 times time points. HBF means hours post-fertilization, and we wanted to see whether there are differences between the yolk area and the muscle area. And you see, independent of time, there are different kinds of spectra available. And then we looked into the idea, is there any change between the time if we only look into muscle area, muscles, or if we look into yolk. And what we found is between muscles of 24 hours and 72 hours, there is indeed a difference. And if you look into the images, you can see that there's formation or the change in formation of the muscles. And if we look into the yolk, after the time, we did not see any difference, which we think makes sense, because why should the yolk change? Yolk is just be eaten up, but there is no change anymore. And we could even find out what was different, and the differences between the young and the old muscles in this case was the RNA formation. The further projects we are planning now in collaboration with the Helmholtz Center in Leipzig is we would now like to take the fish embryos and take different kind of toxicological products from environmental to see how fast Raman spectroscopy can identify changes due to drugs, tox, or environmental conditions. And last but not least, it is also possible to look into the mouse eye. In this case, it's retina image. We have taken an overview with our microscope to see where we are. And then we looked into the area with a 60x objective, and what this we make a screen over it, which means every 10 microns there was one image, to see whether we can overlay now the Raman spectra dependent heat map. That means these colors that you see are only dependent on the Raman spectra peaks that we have at this area. As you see, we see the inner and the outer layer here, getting blue here a little better to see. And the idea was, would it be possible within such a scan to see apoptotic cells, because they are very difficult to see with current methods. And if you look carefully in, we see some little yellow dots. Sorry, maybe I should enlarge this next time. And we assume that these little dots that we see here are due to apoptotic cells. This is a very preliminary test we have done so far, and we would like to continue this proof of concept. So this was just a rough overview I wanted to give to you about how can Raman be used in the 3D environment and with living cells and even living grafts. Especially as we don't need antibodies, we can right away look into the 3D grafts, and we can measure their reaction on any kind of fluidics or drugs when they start to work on the 3D structure. The Raman spectroscopy really helps you to save time, preparation time, costs, antibodies for example, and also it works on only a few material that you need. So we think it's a kind of all-in-one quality test for any kind of cell-based products. And if you would like to prove it, again, I would say just come and see. Thank you very much for your interest and for your time that you took. And if you would like to learn more, this is the lake where we are located. So you will, when you visit us, have a nice environment, as well as you can see us in the next meetings, which would be the EGLD in Düsseldorf, as well as the German Farm Tox Unit in Heidelberg. Thank you very much.
Thanks a million, Karin. I think a few questions came in, and perhaps we can answer a few of these. One is, how is Raman faster or better than FACS?
That is a good question, because usually when people talk about FACS, they say how many millions of cells FACS can analyze during I don't know, a minute or so. Of course, that's what we can do. We analyze a couple hundred cells in time. The problem is, or the challenge is, that Raman don't need any preparation time ahead, and we can really start with a few number of cells, so we don't need to prepare 10 up to 5 million cells before we can get started. So that means it's an immediate test, and you can do it online. That means during culture you can take out the cells, measure them, and bring them back to the incubator and follow them later on. And this is a very fast time to result.
Okay, perfect. Another question was, how fast does an analyzer for 100 spots in the score plot will take? And are you able to make the measurements in an automatic way? So we have installed already automatic.
So there are different possibilities when we work with the system. We can either select singles or the individual cells or spots, pin mark them, pin mark about 50, 100 cells or even more, and let them automatically run. The time for each spot depends on the differences that we expect. If there is a big difference between, let's say, tumor cells and normal cells, then a couple of seconds is good, is enough to measure, 4 to 5 seconds in the moment. If we, however, don't know in the beginning what we expect, we routinely start with about 30 seconds per measurement. And if you would like to do this automatically, there are two possibilities. We can just tell the system with a little, you mark the first cell and the last cell, and you define the steps in between, and then let it run. So if you would like to look into a single cell, then you would take every half a micron or even less a single step. But what we usually do is when we go over the field of interest, like in this retina, or if you would like to measure cells in solution, we can do this also automatically. And then we know about, a cell has about 15 microns, then we make the distance between each spot 15 microns. It depends if we have tumor cells or not. So in this case, what we do, we make statistics. We will run over cells. Maybe we sometimes don't hit one, or sometimes we hit one cell 2 times, but in the statistical field it doesn't matter. And we still can find out where the cells are within, if the cells are not in solution. But if they are fixed, we can go back, because from each step that we do, an image is stored, and we can find cells of interest in the tissue going back into the normal, the original graft.
Okay, perfect. When you make automatic area scans, how precise are they?
I cannot say in microns or in nanometer. I only can tell that they precisely hit. The laser focus is about a micron. So if we start to go into the nucleus, and we start then to look after, I don't know, 100 scans, would go back again, it's a very high relocation reliability, so we would hit the nucleus again. If this was the question, but I'm not sure that I've understood the question correctly. So the relocation of a spot that we have marked is very high.
Okay, another question: is it possible that we can look at the edge of a cell or 2 cells superposed?
Yeah, as we have a confocal Raman system, we usually have a focal size of 1 micron. So if we have 1 cell, we could measure the cell. If the cell is on top, then it depends a little how thick the cell is. We should be able to only collect then this superimposed cells. And of course, also we can focus to the rim of a cell. Then we would have let's say, half the cell, and then you could see maybe are there some exosomes or something like this. That should be possible. We have not yet done this, but from my experience, I think that should work.
Okay, perfect. For the mouse example you mentioned, how do you normalize the spectra?
We always normalize the spectra. That depends on our on the software that we use. And in this case, I have to really make sure that you get the right answers, and I would like to give you to our Raman expert. So this question, it would be better to really answer directly with him. He's not here in the moment. Sorry for that.
No problem, then we will answer this by email. There's another question. Do you have a database to identify your spectra? I think so.
This would be great if we would have it already available, but in the moment, as you know, we start with the system. So for all the cells we have started here, we have measured, we have in the background started to build our database. But of course, compared to the millions of cell types out there, we only started with a small amount of cells. So for example, in the case of the fibroblasts and the keratinocytes of the skin samples or the products, we have already started to build up a database.
Okay, perfect. I think most of the other questions we'll answer by email because there are more coming in. There's, I think, one last question. I think, do you have any literature available concerning the BioRam? I know yes. I think we should perhaps send them also to all attendees, that we have a couple of interesting papers so you can read and follow up what we are talking today about.
And the best thing is really to come and see, because most of the people really in the beginning we don't understand, because we are biologists. Usually Raman spectroscopy was nothing our focus during the studies. But once you see how it works and what you can maybe do with this, then most of the people realize how Raman spectroscopy could help them in their daily routine or in their daily research fields, depending what they would like to do in. So usually you start and then you get more and more ideas during time of learning how helpful Raman is.
Okay, perfect. Then thanks again for the great talks to you, Karin and Heidi, and I will prepare the record and send it to you via a download link together with literature, and you will get the answers for all the questions which came in. Thanks for attending this webinar, and have a great day. Bye-bye.
Thank you. Bye-bye.